Welcome to part 2 video on the protein synthesis or translation series. In this video, I will discuss the mechanism of translation and how selected antibiotics and toxins interfere with this process. In addition, differences between prokaryotic and eukaryotic translation will be highlighted. However, for a better comprehension of this video, I highly recommend that you review the part 1 video on essential components and requirements of translation or protein synthesis. In this segment, I will discuss different steps of the mechanism, highlighting the essential protein factors involved at each step. Protein synthesis is accomplished in the following 3 steps. The first one is initiation, second elongation, and third is termination. I will discuss all 3 steps of mechanism in detail, so let us begin with initiation. As the cell receives the signal for translation, ribosomal subunits are assembled together with messenger RNA aligned with its AUG or start codon to the P site over which initiator tRNA is placed. In this structure, A site is empty and this is known as 70S complex. This is formed in 2 steps. The first step is formation of 30S complex, and second step is binding of 50S subunit with the 30S complex, and this requires the involvement of 3 factors or proteins. They are known as initiation factors. So let us discuss first the formation of 30S complex. Let us discuss 30S complex formation. When the cell receives the signal for translation, 30S subunits bind with IF2, which recruit the GTP source of energy, messenger RNA, which carries information for a polypeptide chain, and initiator tRNA, which is formulated methionine tRNA. This is initiator tRNA, which carries always methionine. This is accompanied with the formation of 30S complex by releasing the IF3. At this point, you should know the function of these initiation factors. For example, initiation factor 1 blocks the entry of initiator tRNA to A site. This is very important. And IF2 actually places the initiator tRNA at P site correctly. Whereas IF3 or initiation factor 3 is an inhibitor for the binding of 50S subunit. Therefore, it inhibits the formation of initiation complex. Therefore, the 30S complex without IF3 is now compatible with the 50S subunit and to form the 70S initiation complex. So 30S complex without IF3 is compatible with 50S subunit. 30S complex has GTP messenger RNA, initiation factor 1 and 2, and the initiator tRNA placed at P site. Initiation factor 1 prevents the initiator tRNA from binding to A site, whereas IF2 places the initiator tRNA at P site correctly. In second step, the 50S subunit bind with 30S complex, releasing initiation factor 1 and 2 and hydrolyzing GTP in the form of GDP and inorganic phosphate as a source of energy with the formation of 70S initiation complex. This complex has P site occupied by initiator tRNA, whereas A site is empty, ready for elongation. So let us discuss now elongation step. Thus, any antibiotic that interferes with the formation of initiation complex or 30S complex will inhibit the initiation step of the translation. The example is streptomycin, which is a type of aminoglycoside antibiotic. This binds with the 16S ribosomal RNA of the 30S subunit, interfering with the correct pairing between the initiator tRNA and the start codon on the messenger RNA at the P site. This incorrect binding leads to misreading of messenger RNA, producing a faulty or non-functional protein and thereby killing the bacteria. Formation of ATS initiation complex in eukaryote is more complex, requires more initiation factors and amount of energy. Initiator tRNA in this case is methionine tRNA. Ribosome requires capside, absolute requirement for binding of ribosome, and there is a scanning for the correct AUG in right context. Right context, I mean this AUG lies in a consensus sequence known as Kozak sequence, which is purine rich region. So let us discuss elongation. In this step, chain elongates by addition of amino acid one by one through the formation of peptide bond. Addition of each amino acid involves four steps. In the first steps is the transfer of amino acid tRNA to A site. Second step is peptide bond formation at A site. Third step is to move the ribosome over the messenger RNA in such a way as to align A site to the next genetic code and peptidal tRNA to P site and uncharged tRNA to E site, from where the uncharged tRNA is thrown out of the site. That is the step 4. These steps require elongation factors and enzymes, and we will discuss these steps in detail in the next slides. So elongation requires a number of protein factors and enzymes. These are known as elongation factor. For example, in bacteria, transfer of amino acid tRNA requires two factors, Tu and Ts. Peptide bond formation requires peptidyl transferase activity of ribosome 23S ribosomal RNA. Translocation requires elongation factor G or translocase, which is energy dependent step. Similarly, in eukaryote, transfer of amino acid tRNA requires eukaryotic elongation factor 1. Peptide bond formation requires peptidyl transferase ribosome activity of 28S ribosomal RNA, and translocation requires eukaryotic elongation factor G or translocase, which is energy dependent step. So let us review the role of Ts and Tu in transferring the amino acid tRNA to A site. As I mentioned earlier, the amino acid tRNA is highly reactive molecule. This is the precursor of translation. Therefore, immediate requirement is to stabilize this, and that is done by factor Tu with GTP. This is active form. It binds selectively, preferentially with this tRNA and stabilize it, and then it try to place it with the ribosome A site. When the amino acid tRNA has been placed correctly at A site, only then GTP is hydrolyzed to GDP, forming this inactive precursor of TU and product of TU, and this ribosome will continue performing elongation. Now the next factor, Ts factor, is required to regenerate this active form from this inactive form. So basically Ts will bind to this, forming Tu-Ts complex, which in turn bind to GTP, regenerating the active form of Tu-GTP. And this is basically in this cycle, Tu-GTP transfers amino acid tRNA to A site correctly, whereas Ts it regenerate the active form of Tu from inactive form GDP-Tu. So second step is peptide bond formation, which is formed at A site between two amino acid, one at P site, other is at A site. So A site now contains peptidal tRNA. This is catalyzed by enzyme called peptidyl transferase, which is a ribosome activity of 23S ribosomal RNA in case of prokaryote and 28S ribosomal RNA in case of eukaryotic system. This step does not require energy, so it is energy independent step. So the third step is translocation, which is movement of the ribosome over the messenger RNA, which is catalyzed by EFG in an energy dependent way, utilizing GTP as a source of energy. In doing so, the peptidyl tRNA is moved to P site, uncharged tRNA is moved to the E site, and A site is emptied here, aligning with the genetic code on messenger RNA. Thus, it allows the continuation of translation. And there are a large number of antibiotics and toxins such as diphtheria which inhibit this step, causing premature termination of translation. So addition of each amino acid requires four steps. Step 1 is transfer of amino acid tRNA to A site. Step 2 is peptide bond formation at A site. Step 3 is translocation to vacate A site, aligning with the next genetic code and moving the tRNA peptidal tRNA to P site, and step 4 is removal of uncharged tRNA from E site. So these steps or cycles continue until ribosome aligns its A site to one of the stop codons UAG, UGA or UAA. There is no tRNA for these stop codon, and therefore they serve as a signal for termination. So termination requires three release factor RF1, 2 and 3 and one of the stop codon. For example, here UAG is shown. When the ribosomal A site is aligned with UAG or one of the stop codon, then it is a signal for termination. In that case, release factor 1 or 2 depending on the stop codon here will occupy the A site, thereby releasing the polypeptide chain, which is functional and active. It start working, and this complex is then disrupted by release factor 3 into its component tRNA, small subunit, large subunit, release factor 1, 2, 3 and messenger RNA, which is hydrolyzed. The initiation factor 1 and 2 binds to the small subunit, and the function of these I have already explained in detail. So this is about termination. Post-transfer quality control is a proofreading activity that takes care of any misincorporation of any amino acid that may happen during elongation or termination. So let us discuss the misincorporation at the step of termination. So this situation where UAA stop codon aligns with A site, it is a signal for termination. However, because of certain reason, unknown reason, the amino acid tRNA has entered here, it has elongated the chain. However, when the chain is transferred to the P site, then release factor 3 releases this wrong faulty polypeptide, which is removed by hydrolysis. This is called as post transfer quality control, and this is conducted by release factor 3. So in summary, there are three levels of proofreading activities which happens. The first one is at the level of charging of tRNA by a mechanism called double C mechanism. We have discussed that in detail. And the second is at the stage of transfer of amino acid tRNA to A site, and this is indicated by hydrolysis of GTP. And the third one is post transfer quality control. This takes care of misincorporation of amino acid into the chain at any stage, including elongation and termination, and this is taken care by release factor 3. Thank you very much for watching this video. I will request you to like it, share it, and subscribe it. Until next time, you take care. Bye bye.